B and T cells have different functions. B cells respond to free antigens, or those on the surface of organisms that circulate outside and between cells of the body. This includes most types of bacteria. However, they cannot recognize antigens located inside cells, such as viral proteins, or certain bacteria, such as meningococci and mycobacteria, which have adapted to live within cells and therefore make detection by the immune system more difficult. B cells manufacture antibody. However, most antigens do not stimulate B cells to produce antibody without the help of T cells. The response to these antigens is therefore referred to as T cell dependent. Unlike B cells, T cells can recognize intracellular antigens, provided they are expressed on the cell surface. T cells do not manufacture antibodies, but they do secrete cytokines which influence other cells. The humoral or antibody response. B cells circulate with a molecule of a three-dimensional protein called antibody on their surface. The antibodies, also known as immunoglobulins, have antigen binding sites where the protein molecules are folded in such a way as to form a three-dimensional cleft into which only antigens of a corresponding shape can bind. There is also a binding site for macrophages and neutrophils. When one of the antibody molecules has a surfaced receptor with exactly the right shape to recognize the antigen, it binds to it like a lock and key. The B cells then enlarge considerably to become large plasma cells, which are antibody manufacturing cells capable of producing up to 100,000 antibody molecules a minute. The antibody molecules they produce have receptors with the same shape that recognize the antigen in the first place. This is known as the humoral response. The first time an infection or vaccine antigen is encountered, the antibody produced is called immunoglobulin M, or IgM. IgM circulates as 5 molecules bound together with a total of 10 binding sites for rapid and effective binding to antigen. When an antigen binds to an antibody, there can be 3 outcomes. Firstly, if the antigen is a toxin or a protein, it can be immobilized and effectively neutralized. Or a macrophage or neutrophil can attach and engulf the antigen-antibody complex. In addition to this, the antigen-antibody complex may activate the complement system. The complement system is a cascade of proteins, some of which are capable of destroying the pathogens. Cell-mediated immunity. When cells contain intracellular antigens, a bit of the antigen is carried to the cell surface using molecules that are part of the major histocompatibility complex, or MHC. T cells can recognize a combination of the MHC molecule and the antigen. When the T cell binds to the MHC antigen complex, the activated cells enlarge, multiply, and secrete cytokines and other toxic molecules, which can then affect other immune system cells nearby. There are various types of T cell. Among these are those that can destroy an infected cell, known as cytotoxic T cells. Another sort, known as helper T cells, can help and stimulate B cells to produce antibody. When an antigen binds to the antibody receptor on a B cell, a bit of the antigen is also taken up into the cell and is then presented to the B cell surface by an MHC molecule. This MHC antigen complex is recognized by a T cell, usually a T helper cell, which secretes cytokines. In this case, the cytokines assist the B cells to proliferate to form identical cells producing the same antibody.